Hello, hi. I'm told we have people at this MENDEL lecture who have never been to a MENDEL lecture before because the topic is so interesting. It's a pleasure to introduce Sarah Tishkoff, who's one of the leading human geneticists, especially working on the origin of humans in Africa and movement out of Africa, and especially focusing on very old human populations in Africa. So Sarah is at the University of Pennsylvania. She's a member of the National Academy of Sciences and the American Academy of Arts and Sciences, I think. And she is originally mostly from California and graduated, partly anyway, graduated from Berkeley, initially with an MA in anthropology, but Berkeley had a number of human geneticists, Alan Wilson and Vincent Sarich and Marie-Claire King, who developed the molecular clock idea and showed that humans separated from apes only about 5 million years ago. So she got into genetics and did a second double degree in genetics, and then went and did a PhD at Yale and on human genetics, again, sequencing African populations. And then she was with Svante Pääbo in Germany for a year, and also in South Africa for a year during the PhD and the early stages. And then she became a faculty member in University of Maryland, and then in University of Pennsylvania. So I'm going to break it off there and present you with the medal. I haven't read a whole Wikipedia article, but
Oh.
that's your medal.
Oh, thank you so much.
That's your
I'll hold it up.
frameable.
Hopefully I don't drop it. Hold on. There we go. All right.
So
Thank you so much. Let me put this down. Okay. So I am truly so excited to be here. I mean, I just can't believe this, where Mendel was doing all of his experiments. This is like for the geeky scientist, this is like nirvana or something, so I'm really excited to be here. And so, of course, you'll recognize on the upper left, this is Charles Darwin. And it was he came up with the model theory for evolution based on natural selection. And then, of course, we have Mendel on the right and developed models of heritability, inheritance as we think of it even today. And it was funny because I was going to tell you originally, I had looked this up and thought that I knew they were roughly contemporaneous, but I thought they didn't know about each other. And then I just walked in that museum and literally one of the exhibits shows the book from Darwin and says that Mendel was reading it. Okay. But each of them, there were gaps, right? So in Darwin's theory, he didn't know what the mechanism was by which the traits were being passed on from generation to generation. And it was only, we have Wright, Fisher, and Haldane here at the bottom, and eventually using mathematical models developed what's often called the new synthesis, and which became the basis for quantitative genetics, showing that traits that have a continuous distribution, like height, could still be explained by, according to Mendel's laws, when you consider that they are influenced by many, many, many genes, and actually we now know thousands of genes are influencing height, each of which, though, is showing, follows Mendel's laws. And so then eventually that led to the modern synthesis and led to the field of evolutionary genetics, which is what my research is based on. So now let me start by talking about what I think are some of the key challenges in human genomics research. The first is that we need to do a way better job characterizing both genomic and phenotypic variation in populations across the globe. We want to understand what the evolutionary processes are that generate and maintain that variation, and to understand how gene-gene, gene-protein, and gene-environment interactions contribute both to normal variation and disease risk. The focus of my research is on Africa, and there's a number of reasons for that. One of which is the fact that all modern humans originated in Africa. So these red dots represent the locations of fossils of anatomically modern humans, the oldest of which is dated to about 300,000 years ago. Oh, this is not showing up, don't know why, but there should be a map you can imagine where Africa is and the rest of the globe, and the point is that relatively small numbers of people left Africa, say around 60,000 to 80,000 years ago, could have been in the hundreds, no more than thousands, migrated out of Africa, giving rise to populations across the globe. We now know that when populations, modern humans, left Africa, they ran into, and it turns out they interbred with archaic populations such as Neanderthals and Denisovans, such that somewhere between about 1% to 6% of the genomes of non-Africans are derived from those archaic populations. And so you can imagine that this demographic history has really shaped the pattern of variation that we see today. More variation in Africa, subset of variation outside of Africa. And the other thing that motivates our research, other than wanting to understand more about human evolutionary history, is to understand risk factors for health disparities. So for example, hypertension is more common in individuals of African ancestry in the US, and adult onset diabetes is more common in people of Native American ancestry. This is largely due to social demographic factors and even things like systemic racism and the ability to even get health care. But it's thought that there may also be genetic risk factors as well. And one of the trickiest things is how do you distinguish between the genetic and the environmental factors influencing these health disparities? And yet, as we showed in this perspective piece a few years ago, if we look at the Genome-wide Association Study database, so it's a database where people are looking for genetic associations with disease, about 80% of the individuals that are included in those studies are of European ancestry. If we looked at this today, it's actually gotten worse. I think it's like now 92% or something. And about 10% are of Asian ancestry, about 2% African ancestry, 1% Hispanic ancestry, less than 1% everybody else, and that's hugely problematic. It's going to impede our ability to fully understand genetic risk factors for human disease. It could actually exacerbate health inequalities. Our ability to translate genetic research into clinical practice or public health policy could be incomplete, or even worse, it may actually be mistaken. It could result in inaccurate assessment of pathogenic variants in clinical genomic studies, and one of the reasons for that is that, let's say that a clinician is trying to diagnose something they think is a genetic condition, they sequence the genome, and they're trying to figure out which variant mutation that I see might be causing the disease. One of the criteria that people use to determine if something is pathogenic is how common it is, with the idea being that if it's common, it's unlikely to be bad for you. So maybe in Europeans it's not common, but maybe if you look at other populations it would be common, and then that's actually going to change the classification. And then lastly, recently, people have been trying to use estimates of genetic risk called polygenic risk scores, where basically, for example, let's say we wanted to look at risk for heart disease. We look at variable sites across the genome, you look for an association with heart disease, and then every variant that you look at has either it's associated or it's not, maybe it's even protective. So then you go into someone else who was not part of that study, and you can literally just look at the same variants and count them up. How many of them are associated with risk for the disease and how many of them might be protective, and you give them an overall score. But what we know is that these are almost always done in Europeans because they have the biggest sample sizes. When people apply those scores, try to do this for people of diverse ancestries, they don't work, and they work particularly poorly in people of African ancestry. So it's really, in my opinion, exacerbating health inequalities. So since I'm going to talk about Africa today, I just want to give you a little bit of background information about it. There are over 2,000 languages spoken in Africa today. These have been classified into 4 major language families based on similarities of the languages. So in blue is the distribution of languages that have been classified as Afro-Asiatic. That would include Cushitic, Semitic, for example, that are mainly in northern and eastern Africa. In red, we're showing the distribution of populations that speak Nilo-Saharan languages, mainly in Central and Eastern Africa. Very often, these populations practice pastoralism, so they have cows and they practice dairying. And then the largest, most widespread language family is Niger Kordofanian, also known as Niger Congo, originated in Western Africa. The largest subfamily of those languages are called the Bantu languages, which arose, they think, around the border of Cameroon and Nigeria. So populations in Western Africa had developed, I don't know how long, but I think it was like 6,000 years ago, iron tool technologies and something called slash and burn agriculture. So with the iron tools, they can cut the trees down. Then they burn down the crops, and that actually makes the next round grow better. And they were really successful. So they expanded all the way across Africa to the east and the south and to the west and the south, where they often interbred with local populations. So they really shaped the genomic landscape in Africa that we see today. And then in green, we have populations that speak with the click, like the San in southern Africa, which I can't speak it, but if you ever saw The Gods Must Be Crazy, that's like a different generation, but it was the population that was in there. And really, when I started, what got me interested, actually, one of the things I was interested in, when I was in Cape Town, I went to a meeting about the history of these populations, and I talked to linguists and archaeologists, and I said, what is the most interesting, outstanding question? And they said, go to Tanzania and study the Hadza and the Sandawi, who also speak with clicks, and they've been classified contentiously as Khoisan languages, but they're very divergent, the languages from each other and from Southern Africa. So I want to say up front that none of this work would have been possible without the partnerships that I've had in the past 23 years or so doing research or field work in Africa. So these are my collaborators. They played a central role in this work. And I thought I'd show you a little bit of what it's like to do the field work. So we have mainly been focusing on populations that are in more rural areas. They're practicing more traditional lifestyles. And they are very tough to get to, so we have to have a four-wheel drive vehicle. We have to bring all of our supplies with us. I like this because you get like the inside perspective of how crowded it is and then the outside perspective. And we're really careful, we're really concerned about doing this research in an ethical manner. So that means that the first thing you do is we have to get IRB consent at our university. But then we have to go through usually many rounds of ethical review in each country. Then we have to get a research permit. And even before that, typically, we're spending very long amounts of time. This is my collaborator, Alfred Njamshi. He's a neurologist in Cameroon. Going to the populations, translating into the local language, explaining what we're doing in layman's terms. Why are we doing? What risk are there, if any? What benefit is there, if any? What are the concerns? And it's only after we get community consent and then ultimately individual consent that we proceed. We also think it's really important to return results to populations, and I don't mean necessarily individual results, I mean like results of the study. And I think it's a form of benefit sharing, and it also builds trust because they know what's happening with the data that they, the samples that they provided. And the other thing that's really important is training and capacity building in Africa, and I've had the honor of having a number of graduate students and postdocs from Africa. Right now, Tafadzwa just recently joined the lab a few months ago. I'm really excited to have her because the idea is we want people to go back and build up the resources and be able to build capacity to do the research there. So we are typically getting blood, and from blood you can get DNA, you can get RNA, you can get plasma. We have to freeze those, the latter two, very quickly. So we have to bring liquid nitrogen with us. And then we also get fecal samples to look at the gut microbiome, urine samples. We get detailed ethnographic information, any information we can get about diet, any medical information they may have, which is usually pretty scarce. And then we have to process the samples in regions where there's typically no electricity. And so nowadays, in the lower left, it's kind of out of the frame of the picture, but we have a generator here. And we can bring that generator and then just set up the lab in the bush. And the types of phenotypic variation we can look at are things that you could do in these really remote areas where you're not in a clinic. And that would include very detailed anthropometric measurements, height and weight and limb length and grip strength and skin color, and cardiovascular lung and blood phenotypes like blood pressure, for example, hemoglobin levels, and so on. We look at metabolic function, lactose tolerance. We're going to soon be doing glucose tolerance tests, and infectious disease status when we can. And what I want to start with today is telling you about a recent study came out last year in Cell, and I must say I really love this image that was actually done by the wife of a faculty member at Penn. And so this was one of the largest studies of high coverage whole genome sequencing in ethnically diverse Africans. So it was 180 individuals from 12 ethnic groups that are shown here. And you can see, just looking at the people, there's a lot of phenotypic variation, there's cultural diversity, there's linguistic diversity, there's quite a bit of genetic diversity, which is shown here. So this is something called an admixture plot. And basically, you're using a probabilistic approach to infer genetic ancestry. So this is made up, each line as a person, and then they could have ancestry from different ancestral populations that are indicated by the different colors. So what you're seeing here by all these colors is how much variation there is in Africa. Tremendous amount, more than we see in most other regions of the world. And then the arrows represent the populations that were included in the study, and you can see that they kind of represent the different colors that are present there, so representing a lot of the genomic diversity. So we found over 33 million single nucleotide polymorphisms, so regions of the genome that are variable. And of those, at the time, about 17% were novel, which is amazing because that's only 180 people. We're now sequencing many more people. We're going to find a lot more variation. And of that variation, a lot is predicted to be functional. So most of it is in non-coding regions, but some are altering protein sequence as well. We can make a phylogenetic tree from this data. And so at the tips of these branches would represent genomes. And then we've given the ethnic group name, and then we've color-coded it based on the language that they speak. And we can see that the populations that have the most basal lineages, sort of towards the root of this tree, are the Click-speaking San populations from southern Africa, followed by the Central African rainforest hunter-gatherers. These are two Bantu-speaking groups who originated in Western or Central Africa in this case. So the Herero now live in Southern Africa, but they migrated not too long ago. And then the Hadza and the Sandawe are those other Click-speaking groups that are in East Africa. And then we have a number of Ethiopian populations in the Fulani who are nomadic, and they're sort of across Sudan all the way Central Africa to Western Africa. And then here are non-African populations. And this looks like what we would expect, right? With this African origin for modern humans, and then small numbers migrate out of Africa. The other thing we did is we looked at this database called the ClinVar database, and that's where people will put what they think are pathogenic variants, so disease-causing variants in there. And remember I told you that one of the issues is that, maybe if you look at more populations, it would change what you're calling pathogenic. So these are just a few examples where we found 44 predicted pathogenic variants that were very common in at least one African population. So here's one that's associated with orthostatic hypotension, very rare in non-African populations, and then this is African Americans right here, but common in the Fulani population. And these are 3 amino acid substitutions that this gene, PAD, I think that's an I3, associated with a type of alopecia that's very common in patients of African ancestry. And we can see that these variants exist in the African American population, but there are several groups that have these at a very relatively high frequency. So does it mean they're not pathogenic, or could it be that there are other modifying mutations? We don't know, but it's something to look into. We then use a measure of heterozygosity. We want to see how much variation is there in the genomes of these African populations compared to the reference genome, which is predominantly European, maybe some parts African American as well. And so we see that the populations that have the most diversity are the two click-speaking San populations, followed by the Central African rainforest hunter-gatherers. At the lower end are the Amhara from Ethiopia, and we know that there's a lot of historic migration and admixture in that population. And that may explain why they're at the lower end with non-Africans, I should say. And in fact, if we were to look at non-Africans, it'd be somewhere just below here. So Africans have a lot of variation. We then use coalescent modeling, computational approaches to make inferences about changes in the effective population size going backwards in time. This particular approach, this PSMC, is only informative for older events, so 50,000 years and going back. But what we see is that in the past, it was the hunter-gatherer populations, the San and the Baka rainforest hunter-gatherers, that had the largest population size. Now, this other approach we use, SMC++, is more informative for recent events. And here we can see that now these groups like the Amhara and some of the Bantu-speaking groups are much bigger, their effective population size. But some of these groups, particularly these two hunter-gatherer groups, the Hadza and the Shabu, they only have a census size each of about 1,000. And they had a dramatic population crash, we think, in the last few hundred years. And then groups like the Fulani also, pretty low effective population size. And then using a number of different approaches together with our collaborator Yun Song, including approaches that take into account more complex demographic models. So I showed you that phylogenetic tree, but the problem with that tree is, let's say two populations interbred together, they admix, and now they're gonna come out in the middle of that tree, right? And so it's not taking into account migration and gene flow. So in this model, we take into account migration and gene flow and population size. And what we find, unlike other models of modern human origins, is that we think there was an ancient shared common ancestry between the Central African rainforest hunter-gatherers and the Southern African San. They diverged about 80,000 years ago. And it was the ancestor of these populations that gave rise to all other populations. The other thing you see is this thing called a ghost, and it's not a ghost like you think of a ghost, but it means a population that doesn't exist today, but we infer it from the model. If you put this into the model works better. And this would have been a lineage that would have diverged around the time of Neanderthals and modern humans. Okay, so imagine it kind of like Neanderthals, but in Africa. And we think that there was intergression. They were interbreeding with modern humans prior to the divergence of all modern humans, and then more recently in certain populations. The other thing that you see, I'll point out a couple of other things. So one, and this is out of Africa, so these are non-Africans. You see that the divergence times of all these populations is pretty old. So there's been, we call it substructure in Africa for a long time. And then you see these blue lines, they represent migration events. So we can see, for example, the migration of the Bantu speakers, and then they admix with other people. This was interesting because we saw strong evidence for admixture between the ancestors of the click-speaking East Africans and the ancestors of the click-speaking Southern Africans. Could possibly explain some of the similarities in their language. So the other thing that we're interested in, I want to focus on for the rest of my talk, is how people have adapted to different environments and different diets. So this is from a review paper giving some examples. So some things you might have heard about, for instance, is sickle cell. So the sickle cell mutation, when it's homozygous, you have 2 copies of it, is deadly, particularly in an African environment. Typically, children who have this may die by age 5 or so. But if you have 1 copy of the sickle mutation and 1 unaffected copy, you're protected against malaria. And so for that reason, it maintains a high frequency in populations where malaria is prevalent. So, and there are other examples. Lactose tolerance is an example where different mutations arose in Africa and in Europe. So one of the ways that we are looking for selection for local adaptation in particular is we use something called an FST statistic. So it's just a way to look at allele frequency differences between populations. And we're using something called the DI statistic. So this to demonstrate, let's imagine we were going to compare a Baka hunter-gatherer to a Takari agriculturalist and a Mercy pastoralist. And we do, for every variant, you calculate the pairwise FST between all of these different pairs of populations. And we're looking for variants that are very different frequency, specifically, let's say, in the Baka, but they're more similar, these two groups are more similar to each other. We get thousands of them. You get a distribution, and so we take the top 0.01%, and then we say, and there's 10,000, and then we say, what are the genes that are nearby, and what do those genes do, based on looking at like gene ontology databases. And that might give us a clue about different examples of local adaptation. So I'm going to highlight a few today. The first is in the rainforest hunter-gatherers, excuse me, from Central Africa. And these populations are characterized by short stature. It's thought to be an adaptive trait to a tropical environment. And what's really interesting is that when we look at regions of the genome that are different in those groups, we see enrichment for genes that play a role in bone development and chondrocyte differentiation. So now we want to try to identify what those are, and they probably play a role in stature in that population. Now let's look at a different group, the Hadza-Click-speaking hunter-gatherers in Eastern Africa. They live in a very different environment. And here we see a striking enrichment for genes that play a role in heart muscle development. And you could imagine how that might be adaptive in these populations that are walking very long distances. And so I have a researcher now in my lab who's following up on these. Many of them they're almost all in non-coding regions, and we think they regulate expression of genes that play a role in heart development. And that could give us a clue. In a way, maybe these guys have like super hearts, like super well-functioning hearts. And we could understand more about heart development and also what goes wrong in some people and causes disease. Next, I want to tell you about a study that was led by a postdoc in my lab, Mike McQuillan, that we published in 2022. And here we use what's called a SNP array. So we're not doing whole genome sequencing. We're taking known variants, and we're looking at their frequency in over 1,000 Africans, and then we do this admixture plot, and you see all the diversity. And these, as you can see, from number of regions in Africa. And then we're going to look for genomic footprints of selection. So here's one common test. I already told you about one to look for local adaptation. So now I'm going to tell you about another one. So this one, imagine that these different dots represent variation in this region of the genome. And let's say a mutation arises that increases the fitness of the individual who has it, which means that they have more kids and their kids have more kids and so on. And it's going to rapidly rise in frequency in the population. When it rises rapidly in frequency, it drags with it the neighboring variation. So everybody who's homozygous here for this mutation that's under selection is homozygous for the yellow variant and the purple variant, and you can keep looking, going long distances, and that's what they call extended haplotype homozygosity. So what Mike did is he did this DI statistic, and he did what's called an IHS test for looking at this extended haplotype homozygosity. And we look at the top 1% of variants, and then we do this we look at the genes that are nearby, and we ask what are they doing to identify biological functions or pathways that may be under selection. So there was one thing that stood out to us, and I know you can't read any of this, so I'll tell you what it is. In many of the Ethiopian populations, we saw enrichment for genes that play a role in alcohol dehydrogenase activity. And this was just kind of unusual. We don't usually see the shared signature of selection in so many different populations and being so specific to that region. So alcohol dehydrogenase gene family encodes enzymes that catalyze the metabolism of ethanol into the toxic intermediate acetaldehyde. Non-synonymous, so protein-altering mutations in the ADH genes, including this very well-characterized variant called ADH, I'm going to just call it ADH1B. Anyhow, this particular variant increases the catalytic activity of the enzyme, and that can lead to a buildup of acetaldehyde. This mutation in a number of genome-wide association studies has been shown to be protective against alcoholism and is highly associated with alcohol drinking behavior. This particular variant is very common in Asia. It's been shown to be a target of natural selection. It's associated with the flushing response after drinking alcohol. And it had been hypothesized in other studies. I think they estimated in Asia, if I remember, it's like maybe within 10,000 years was the age of the mutation. And they thought that it had to do maybe with the advent of rice agriculture and the associated increase in fermented foods and beverages. So in Ethiopia, they also have a lot of fermented products that they are consuming. But this also plays an important role in a number of metabolic pathways, right? So we don't know what the selective force is. This is just showing that in a subset of the Ethiopian populations, those who specifically practice agriculture. So this is what was kind of cool, was that we only see the signature in groups that practice agriculture. So here's the ADH1A gene, and we see over hundreds of thousands of nucleotides, the signature of local adaptation and a signature of a selective sweep. Now, if we look at a population from the same region, the same genetic ancestry, but they don't practice agriculture, we don't see a signature at all. So this is in this region, there's a haplotype that becomes really common, and nearby there are other genes of interest, okay? So it's not like it's only that one gene. There are these others that are nearby that also show a signature of selection. We could see in red, that's showing the frequency of the variants that we think may be targets of selection, very common just in these Ethiopian agriculturalist groups. Here's that signature, that extended haplotype homozygosity that I was talking about, which you see in all of these. So these other genes, ADH7 is involved in both ethanol and retinol oxidation. MTTP plays a role in lipid metabolism. So these are also important. If we look at, this is called a haplotype network. So a haplotype is how different variants are arranged along some short segment of a chromosome. And then these circles represent the haplotype, and the size of the circle represents the number of observations of it. And different colors here represent the prevalence in different populations. And here, this is the same exact network, but now we're talking about who has that derived mutation I told you about that's associated with flushing in Asians. Here's the East Asian haplotype. The one in Ethiopia is highly divergent. So there's no way that this was introduced by migration. This arose independently. Now, it turns out it is actually at low frequency in Druze from, say, the Arabian Peninsula, and also in Europeans. And in fact, we think that it was introduced by migration from those populations into that region. One of the reasons we think this is we can do something where we look at local genomic ancestry. So right here, we're looking on chromosome 4. This is where the ADH region is. And what we're doing is we're sliding along the chromosome, and for each person, each row is a person, we're saying at this position, do they have African ancestry in purple, or do they have European ancestry in yellow? And what we see is an enrichment for European ancestry right in that region. And it's more extreme than what you see in the rest of the genome. So we think this was introduced by historic migration and admixture. We use this method to estimate the timing of that admixture event to be around 2,000 years ago, and this also fits with historical documents of migrations into that region. And this is showing, if you look at the frequency of that variant, there's a significant difference between people who practice agriculture, where it's very common, and people with the same exact genetic ancestry, but they practice different subsistence patterns, hunter-gatherers or pastoralists. It's very rare. So in conclusion, there are multiple putatively functional variants in that region that impact alcohol metabolism and possibly lipid metabolism that show signatures of selection. These were likely introduced from a West Eurasian source within the last 2,000 to 3,000 years. And these data support the hypothesis that agriculture has shaped the evolutionary history of this region, but we still don't know exactly the selective force. All right, last example is going to be skin color. So it is thought that skin color is an adaptive trait. So these blue dots represent the levels of melanin. Melanin is the dark pigment in skin that is produced in melanosomes, which are within melanocytes in your skin. And it's long been noted that there's a strong correlation with UV levels. So it's thought that when our ancestors left Africa and went to places of higher latitude and less UV exposure, that there might be selection for lighter skin, possibly because it would promote vitamin D synthesis, which occurs in response to UV. It's actually synthesized in the skin. If you have a deficit, that could be really problematic. Whereas close to the equator, there would be selection for darker skin, which is protective against sunlight, also may prevent folate degradation as well. So to measure skin pigmentation, we use a spectrophotometer. We shine a light under the arm because that's a place where you're not going to get a lot of exposure. We also did it on the wrist. Some people do that to look at like tanning response. And based on the wavelength of the reflection, you can infer the melanin levels. And we see a big range of skin colors in Africa. And the most lightly pigmented people are those click-speaking San, who I told you have some of the oldest lineages in Africa. This is not due to recent admixture with non-Africans. And then we have a whole range of variation. And then we have the most darkly pigmented people in the world, actually, from South Sudan. So, back in 2017, we published this genome-wide association study with a pathetically small number of people. People who do these GWAS typically look at tens of thousands of people. We looked at 1,500 people in Africa because it's hard to get this data. But we still found 8 variants that were independently associated at a very stringent genome-wide level of significance in 4 regions of the genome. So I'm just going to quickly step through. Strongest association is a non-synonymous variant that alters the protein of SLC24A5. This was actually already known to play a role in skin color in Europeans, and was actually the very first gene identified to play a role in human skin color. And here in these plots, the blue always represents the light-associated variant, and we could see it's at almost 100% frequency in Europeans. It's common in Pakistan, some parts of Southern Asia, and then very common in East Asia. And we wanted to know, did it arise independently, like that other variant I told you about, or was it introduced by migration? And here we can see that there's this one very long common haplotype that we see this variant in Europeans. And in East Africa, the variant is on the same haplotype background. So that means that it had to have been introduced by migration, we think, into Africa. And then it was subsequently under selection in Africa. The second strongest association is at a gene that at the time had no name. It was just called open reading frame, whatever, and then it got a name when we were in the middle of the study based on homology to iron protein transporters, iron transporter proteins. And so MFSD12, we found 2 independent associations, one in an upstream regulatory region that's regulating expression of MFSD12. The other is within the MFSD12 gene. There is a non-synonymous variant, but we really don't know for sure what those variants are doing. And here we can see that the variant upstream that's associated with light skin color is common in Europe and in Asia. It's common in East Africa, and it's very common in the San. Now, what's it have to do with pigmentation? So our collaborator Bill Pavin, knocked it out using CRISPR technology in a mouse, and it had a dramatic impact on pigmentation. And so now many people have now confirmed this, and it's also been shown to play a risk for melanoma. And what I find really exciting about this is that even with people say, oh, you shouldn't study minority populations because you're not going to have enough samples to do anything, but we found a novel, never before described gene. We're actually the first ones in PubMed, if you look, with 1,500 people. So it's possible. And particularly in African populations where there's so much genetic and phenotypic variation. With our collaborators, Yun Song and Ethan Jewett, they look at the gene genealogy. So just think if you unwrap this, it kind of looks like that tree. But at the end here again are the sequences. And here an open circle means that there are sequences that have the light allele, and these have the dark allele, and then we have the different populations. Now in this case, the ancestral allele is a light allele. So dark is more has been derived, but it's still really old, almost 1 million years old. So light and dark alleles have evolved in Africa. So the third locus, there were again 2 independent associations in non-coding regions. We thought that the most likely candidate was DDB1. It plays an important role in DNA repair after exposure to UV. Mutations in this gene or in the proteins that interact with this gene cause xeroderma pigmentosum. Children can't be exposed to sunlight because they can't repair the DNA, and they get skin cancer. There was some preliminary data at that time that suggested there may be interactions between these variants and that gene. And here, again, if we look at these upstream variants, they're very common in non-African populations, the light-associated allele, and then common in some of the African populations. What does it have to do with pigmentation? We don't know, but we were intrigued that when I looked up the gene, I see that it is the gene that causes the color of tomatoes. So it's doing something with pigmentation. I'm going to come back to that later. Okay, so now I'm going to tell you your third lesson of test of natural selection for the day is Tajima's test. And this one is based on something called the allele frequency spectrum. So what that means is here you're looking at the number of singletons in a population, the number of doubletons, tripletons, and so on. This blue line is what we expect under neutrality. Because of our demographic history, we expect more rare variants, for example. But when we see positive selection or population growth can cause an excess of rare variants beyond what we would expect, or balancing selection will cause an excess of common variants. Okay, so remember, a negative value is going to indicate positive selection. And what we see, this is European population from 1,000 genomes. This is Chinese, this is West African, Nilo-Saharan, and so on. Over hundreds of thousands of nucleotides, we see this very strong signature in the Asian and European populations of positive selection. And it's more striking if you look at the heterozygosity, the variation in the region. It is wiped out in Asia, like just completely wiped a variation in that region. Lower in Europe, and you don't see that at all in Africa. And if we look at the haplotype, this is over 200,000 base pairs. There's this common long haplotype in non-African populations. And if you look at the gene genealogy, this rapid coalescence right around 60,000 years ago, the time of migration of modern humans out of Africa. So we think that when this was introduced out of Africa, there's something in that chromosomal region, and it may not be these variants at all, maybe something else, is so important that it rapidly swept through the populations. A very rare example of a selective sweep outside of Africa. Okay, so then the fourth look wasn't a huge surprise. OCA2, already known to play a role in pigmentation, and in fact, mutations in OCA2 are associated with albinism. We found some variants in this upstream region and this other gene that are non-coding, and they're influencing expression of OCA2. There are mutations here that are associated with skin color and eye color in Europeans, but these are different than those. And then there's a synonymous variant in exon 10, and it's not associated with gene expression, but it is associated with alternative splicing. And the variant T that's associated with light skin is associated with a shorter transcript. And that transcript is missing exactly exon 10, but it's still coding for a protein. It's still in frame. But that protein is missing the third transmembrane domain completely. So that's clearly going to have a major impact on the protein function. So in what we found is that in half of the cases, the ancestral alleles, the light allele, not the dark allele in Africa, and most of the derived mutations predate the origin of modern humans. What does that tell us about the evolution of skin color? Well, this poor chimp lost its body hair, and you can see it's relatively lightly pigmented. I've been told not all chimps are like that. There may be some variation, but anthropologists thought that that is the ancestral state because you wouldn't need dark pigmented if you've got fur, right? And when our ancestors left the forest and went into the savanna, there would have been selection to lose the body hair for thermal regulation purposes. And if you lose the body hair and you're out in the savanna, there'd be selection for darker skin. So when you look at these reconstructions of populations in Africa, they're always darkly pigmented, but we've shown that both light and dark alleles have been evolving in Africa. So there was probably variation for some time. And the other really cool thing was that the only other place in the world where we see the dark associated allele outside of sub-Saharan Africa is South Asia and Australo-Melanesia, two regions where we see darkly pigmented people. So we were able to trace these back to Africa in these particular cases and show that they were introduced by the migration out of Africa and then were maintained in those regions due to natural selection. Okay, how much time do I have left so I just know? How much, 15? we might be able to just zoom through. I'm sorry, I might be a few minutes late. But okay, last example. Now, the southern sun, we did that test of selection I told you about. And here we see enrichment for genes that play a role in pigmentation. Also, interestingly, hair follicle morphology. It's interesting because they have a very unique hair type, this very tightly wound hair that occurs kind of in patches. It's very unique. And narrow eye opening, another morphological feature that is different in that population. So then in this paper we just published recently in Nature Genetics, Huan-Chen Feng, a very talented postdoc in the lab, decided to repeat the GWAS, but he just used imputed variants. So let's just say there are more variants, but the results largely the same. But then we said, we don't care if it's genome-wide significant. We're going to just take like the top 5,000. And when you take the top 5,000, you see major enrichment for genes that play a role in pigmentation, meaning that there's biologically informative information even if they're not at genome-wide levels of significance, which we can't get with small sample sizes. So what he does is he takes the GWAS hits and he takes the selection scan hits for the sun, and then we have tens of thousands. And then he looks for ones that are in predicted open chromatin regions in melanocytes, that's the relevant cell type. So we narrow it down to 1,157. We use something called a massively parallel reporter assay where we are making these oligos that have either the dark associated or the light associated allele. You put it into this lentiviral vector, which has this barcode. You're infecting melanoma cells, and you're essentially doing like an RNA-seq analysis where you can look at differential expression between the light and the dark allele. So what we want to find are variants that show a big difference between the light and the dark allele. And we did this in darkly pigmented skin, and we did this in lightly pigmented skin. And what's interesting is you get different results. So about 45 overlap that we see a significant difference in expression, and overlaps at about 45. But some are specific either to the lightly pigmented cells or to the darkly pigmented cells. So in other words, sort of the genetic background matters. And here's an example at a gene called MID-F. It codes for a transcription factor that's known to play a role in melanosome function. And here we have this variant, though, that shows a signature of selection, and it's only common in the sun. So the ancestral allele C is high in the sun. And these, we found actually some variants here, the variants that are under selection. They happen to overlap these predicted enhancer regions. And then we generated high C and H3K27 acetylation high chip maps to look at chromatin interactions. And we can see that these enhancers are interacting with this shorter transcript that is only expressed in melanosomes. Okay, so by doing these maps, we are able to show what the target genes are of these regulatory variants. And then we use CRISPR, either CRISPR inhibition, which is going to, the CRISPR Cas9 inhibitor binds to the enhancer and represses the activity of the enhancer and promoter and reduces mRNA transcription, or we just knock out the enhancer, just chop it out using CRISPR-Cas9 method. So we're going to, for example, focus on this region where the predicted enhancer is, and what we can see is that when we do the CRISPR inhibition, and let me just see which one is this one. This is the CRISPR inhibition. We see a decrease in expression of MID-F. And what's kind of cool about this is you can actually look at the cells and see if the color of the cells changes. So we look at the darkly pigmented, and we see that the variant associated with lighter skin in the sun is showing lower pigmentation levels. So now we can show that it's actually having an impact on the phenotype. And then we do this for the knockout, and again, you see knockout the enhancer, decrease in expression, and impacts the phenotype. We can then, after, for example, we do the knockout, you can look at gene expression and see that there's altering the expression of a number of genes that play a role in pigmentation. This was one that I thought was kind of cool because this was found from the selection scans. This variant is at almost 100% frequency in the sun. It's at almost 100% frequency in most non-Africans, except for Oceanian populations that are darkly pigmented. And this dark associated allele, or this variant, there's this other variant that's in most of sub-Saharan Africa, If we compare populations from 1,000 genomes from Europe or from West Africa, it's among the most differentiated in the entire genome. And we see this predicted enhancer region. Again, we can show that the C variant is associated with decreased activity, and when we do CRISPR-I, again, we see a decrease in the gene expression. And oops, I missed it somewhere, but here we go. The melanin level also decreases. So we show that this is an enhancer. We show it's interacting with this gene TRPS1, and that it is influencing pigmentation, and it's been under very strong selection globally. Now, I want to go back to DDB1 because we took a closer look. This came out as one of our very top hits from that screen. It's near these genes TMEM138 and CYB56183, and I told you we thought this was the target gene, and this is why these experiments are so important, because it turns out we're pretty much wrong. Kind of. It is a target gene, but it's not the major target gene. Because what we find is that, in this case, here's the variant. Now, here, the A variant is associated with light skin, and the T variant is associated with dark, so just keep that in mind. Because here, the dark-associated variant is lower expression. That's the opposite of what we saw before, okay? And we can see, again, that there is interaction, and actually there is interaction with DDB1, but there's a stronger interaction with these two nearby genes, TMEM138 and CYB56183. And here, when we do CRISPR inhibition, we see a significant decrease in expression of those two genes, not significant for DDB1. However, when we do the enhancer knockout, we do see a significant difference for all three. So DDB1 is effective, but just not as strongly, we think. And this becomes more clear because if you take either the knockout or the CRISPR inhibition, the cells where we've used CRISPR inhibition to stop expression, and then do RNA sequencing, you see that the biggest difference in expression, a decrease in expression, is for TMEM138 and CYB56183. We don't even see DDB1 here. And the problem, though, when you knock it out, you don't see a change in the melanin levels, but that's because we're using darkly pigmented cells, right? And I said it's the dark-associated alleles associated with lower expression levels. So the only way to see that phenotype is to overexpress the protein. So that's what we did. We overexpressed the protein, and then we used this control, CD63GFP. It's expressed in melanosomes, but doesn't affect pigmentation. And we could see that when we overexpress this, we see a decrease in pigmentation. So we show that, yes, this gene is playing a role in pigmentation. That was not known before. So again, after the overexpression, we see changes in gene expression for a number of genes that play a role in melanosome function and also mitochondrial function. And this is just showing that it alters the expression of some well-known genes that play a role in, or it's correlated with that, genes that play a role in pigmentation. Now, we were particularly interested in this gene because it's highly expressed specifically in melanocytes, whereas TMEM138 is not particularly high in melanocytes. Our collaborator, Elena Awancia, then looked to see where the protein is located in the melanocyte, and she sees that it is co-localizing with TYRP1, which is expressed in the melanosome. So it is in the melanosome, as we would expect, if it has to do with pigmentation. So what does this have to do with pigmentation? We don't know, but we know that this is a transmembrane reductase. It reduces iron cations in the lumen of the late endosome and lysosome, which are very similar structurally to like melanosomes. So we think that it may be involved in endosomal, lysosomal, and maybe melanosomal cellular iron homeostasis. What does it have to do with pigmentation? I don't know, but when I look up iron and pigmentation, I found some papers, such as this one saying it upregulates melanogenesis in cultured retinal pigment epithelial cells, and people who have intravenous iron infusions then have this pigmentation. So I think there is some evidence that it's maybe playing a role. And we're not ruling out this gene. Turns out it may be interesting as well because it plays a role in development of cilia, and it turns out that cilia could possibly play a role in melanogenesis as well. And in conclusion, we need to study more diverse populations. African demographic history is complex. High levels of genetic and phenotypic variation in Africa are informative for genome-wide association studies. And we need to integrate those with signatures of natural selection, which can be informative for identifying candidate functional loci. But ultimately, we need functional genomics to show what is causal and what is not, and to identify target genes. And with that, I will just thank the many, many people who contributed to this, and I'm happy to answer any questions that you may have.
That's amazing. Thank you. Let's have questions.
Thank you for a wonderful talk. I've got two questions. One is regarding the DNA repair capacity of these different populations, because these are fairly distant from an evolutionary perspective. So I was wondering whether they have the same DNA repair capacity and therefore similar rate of mutation.
No idea. The only thing I could say is, I mean, I'd be surprised if there was a big, this is like pretty critical for all humans, right? And so like, but that's an interesting question, because you might say, well, maybe the selective force is more. We don't know. We really don't know. I mean, that's kind of an interesting question, actually. So yeah, we should think about that. And you had a second one.
So you show data from nuclear genomes, from sequencing of the nuclear genomes. So I was wondering, what is the level of variation in mitochondrial DNA, and whether there are any differences between nuclear and mitochondrial DNA in this respect? So
we actually haven't looked recently at these, but we have another study. We've done this before. We still have to publish this, actually. But many people have studied mitochondrial DNA variation in African Y chromosome. Again, it just reflects that phylogeny, more diversity, oldest lineages in Africa, a subset of that outside of Africa. But there are people like Doug Wallace at Penn who thinks that mitochondria are super important, and we should probably be looking at variation in mitochondria and not just ignoring them and only looking at the nuclear genome or the autosomes, So they may very well be important for some phenotypes, right? I mean, that's possible. Yeah. Okay, who's next? Who's next? Here? Yeah, give him.
Yeah, great. Thank you for a fascinating talk. I would like to ask about the missing heritability problem. Like when I was
the
so-called missing heritability problem.
Oh, yes.
Like when I was doing my PhD with Nick Barton in Austria, we often touched that topic, right? Like you have a GWAS of, I don't know, human height, which includes like hundreds of thousands of individuals. Yet with the significant loci you identify, only explain like a small fraction of heritability, I don't know, 10, maybe 20%, and that's really good.
Yep.
Maybe it has changed because it's been some years ago, but what I wanted to ask, like what fraction of heritability you explain by the loci, like the skin color?
Yeah, I'm trying to remember.
Loci.
I'm trying to remember for the pigmentation. If the number that's coming to my mind, I think it was like 30-something%. I could be wrong. I can't remember. That's what I seem to recall.
Okay.
But the point is that you're saying, so for example, when people do genome-wide association studies, you find all these variants that seem to be associated with whatever trait, but they almost never explain most of the variation. And height should be one of the most straightforward. It's very easy to measure. Almost every gene plays some role, it turns out, in height, but it's still, you take the whole, every gene that's known, it's only, 40%? I think they're up to like 40%, something like that. So there could be a number of causes, reasons. So one is that we are not finding all the causal mutations, right? There are more mutations out there. We don't know about them. There could be epigenetic changes. There could be influence of environment. There could be epistatic interactions, right? That could be playing a role. So gene by environment, gene by gene interactions, and the fact that, certainly for the Africans, it's because we just haven't done enough studies. We don't have a big enough sample size. We need to keep going to find these variants, and that will also be important for these like polygenic risk scores, that they might actually be more effective in populations across the globe.
Thank you.
Anyone else? So if humans separated from chimpanzees 5 million years ago, and say we have 10 base pair changes in every 1,000 differing from chimps, and then you're inferring or inferring ghost populations that are 500,000 years old, Are they recognizably 5 or 10% more similar to chimpanzees?
Well, remember, we don't have the genome sequences. So all we are is we're inferring. It's sort of a population that goes into the model that you infer the amount of overall divergence. And so, you know, I'm just waiting for the day that Svante Pääbo or somebody else is able to sequence a genome, an ancient genome that is that old in Africa. That would be fascinating. The oldest they can get so far, they claim 15,000 years. I don't know if I believe it, but let's say up to 15,000 years. But you're still looking at relatively more recent events. So there was also a fossil, I think from Cameroon, that morphologically they claim that it looked like it could be some kind of like mixture between archaic and modern humans. So there's no reason to think that this wasn't happening in Africa. It was happening around the rest of the world, but we just don't have any ancient genomes, so we just don't know.
So you only have part of a complete genome, even if you take all that you have out of existing ones.
Yeah.
I guess people who are aware of the San not being dark-skinned realized long ago that the lighter skin might be the ancestral African thing. I didn't. But are any of these light skin variants coming through from chimpanzees? Or
So again, I don't know for sure. And I don't know that people thought that. I think most people thought that dark skin, that the ancestral state was dark. But all I can say is for these that we've been looking at, we see that both have evolved in Africa. And as I said, about half the time the ancestral. And then we determine that because we compare to the chimp. So we compare to the chimp and then we say, which one looks chimp-like and which one looks more recently. So yes, for each one, we have one that looks ancestral and it shows similarity to chimps. And we have one more derived, but it's, you know, the derived can be either dark associated or light associated. Okay. And even so, they're old, like they're, you know, hundreds of thousands to a million years old.
more questions?
Yeah, in the back.
Hi, thank you for the nice lecture. My question is like relatively new evolutionary new mutations. How are they fixed into germ cells so that later they can passed and naturally selected and, you know, fixed? So first of all, how the somatic mutations are fixed into germ cells, like transferred into germ cells. For example,
Wait, I didn't hear that. How are somatic mutations what?
How the somatic mutations are fixed into germ cells so that they can propagate to the next generation. For example, mutation in alcohol dehydrogenase, it's somatic, somewhere in the gut or in the...
Well, not the ones we're looking at. I mean, so the ones that we're looking at are from the germ cells, right? It's genomic, it's inherited. So the somatic, you know, by definition, they would be mutations that might be occurring in different cells in the body, but they're not going to be passed on. We can thank Mendel for, you know, understanding that now. So I'm not quite sure what the question is because that particular example, it is passed down through germ cells. Yeah.
Thank you.
More question? I think Mendel would have loved this talk. Thank you.
Sure.